Hello and welcome to the masterclass for psychologists on the mucopolysaccharide doses. My name is Elsa Shapiro and I am the lead faculty for this program. Thank you so much to the National MPS Society and to the various sponsors for their support in making this program possible. The goals of this masterclass are to provide psychologists with the knowledge of these diseases, their clinical manifestations, and the tools for the best assessment of these children and individuals who have the MPS disorders. While we're going to spend some time on the clinical aspects of the disease, um, we're going to have to spend some time on the natural history of these disorders and research and clinical trials because they provide the information that you need for a clinical assessment as well as working within a clinical trial. MPS disorders are now the focus of many, many trials, and that was the impetus for this program. There's gene therapy, the CRISPR, enzyme replacement, gene editing, many different kinds of treatments that are now coming online and are being tested. And so we need to understand these diseases in much greater detail. So what are the mucopolysaccharidoses? Well, they refer to a group of 7 different diseases or conditions that are considered to be lysosomal diseases. What are lysosomal diseases? Well, they are diseases that have to do with the lysosome in the cell, which is the uh, uh, cell that provides the means of breaking down various molecules that are important in overall metabolic conditions. And because they're absent or malfunctioning, there's an inability to break down these long chains of molecules that are found throughout the body. And they're called glycosaminoglycans, but GAGs is a much easier term to use. Over time, these GAGs collect in the cells and in the blood and in the connective tissues, and it results in permanent, progressive cellular damage that affect appearance, that affect physical abilities, that affect all organs in the body and various systems in the body, and in most cases, but not all, mental development. Each of the MPS disorders has a unique enzyme that is absent or doesn't work. Um, there are 11 different enzymes that have been identified in MPS disorder, and each MPS disorder has a unique clinical presentation. Now, there are 4 of these GAGs that are found in the urine of MPS patients, but the one to really think about closely is something called heparan sulfate, because it is found in those diseases which have a progressive decline of cognition. Um, Keratin sulfate is another one of the GAGs, which is found in diseases with skeletal and connective tissue abnormalities. Um, There's also chondroitin sulfate and dermatan sulfate, but we don't need to really focus on these. It's the heparan sulfate which really causes the cognitive abnormalities or is associated with the cognitive abnormalities. We're not exactly sure how that works. Um, Most patients with MPS disorders have a shortened lifespan because of all of the body systems that are involved in these disorders. Um, and they're all genetic diseases. Most of them are autosomal recessive. That means you need to have a gene from each parent in order to um, uh, have the disease. But MPS 2, which we'll talk about later, is an X-linked disorder, which is obviously primarily seen in boys. X-linked meaning transmitted by the mother only. Now, here is a list of all of the MPS disorders. I'm not going to go through them all. You can look at these at your leisure, and I have listed the treatments available, whether or not they have cognitive decline or abnormal behavior. And abnormal behavior we'll be focusing on a lot during this program. Now, the MPS disorders have a lot of brain abnormalities um, and somatic abnormalities. And so, what I have here is the kinds of different direct disease related problems that we see. For example, hearing loss, airway disease, hernias, carpal tunnel syndrome, uh, cord compression, which results in neurologic problems, um, cardiac disease, liver disease, skeletal deformities, joint stiffness, and carpal tunnel syndrome, even in children. Um, there are also indirect effects. So these somatic problems, especially hearing and vision loss and motor abnormality, as well as hydrocephalus, can have indirect effects on cognition. So let me tell you a little bit about the timeline. And this is another set of slides that you can look through at your leisure. Um, the first treatment for one of these diseases began in 1981 in the UK by John Hobbs. And he reported results after a year after bone marrow transplantation on the Griffith scale of mental development. Um, and then in the early 80s, we had some natural history work, Young and Harper, and uh, these uh, started working in MPS 2, and they separated severe and attenuated forms, and then they characterized the decline in MPS 3. In 1986, the first standard protocols were developed at Minnesota to measure the outcome of bone marrow transplantation. And they continue till this day, first using the old Bailey, then we changed to the Mullen, and then to the Bailey back again in 2014. And we added the Vineland in 1992, and we continue to use that in all our protocols at Minnesota. Um, in 1993, we described childhood dementia and the distinction between static and progressive disease. That is, disease that just causes abnormality in cognition versus decline over time. And in 1995, we presented the outcomes of bone marrow transplantation for MPS 1, 2, and 3, MPS being successful, MPS 2 and 3 not. In 2004 was a very important time when the lysosomal disease network started. And that was designed to look at the natural history of rare diseases. And from 2008, we had NIH funding for this, and the first fellow was Julie Isengard, who you will meet later. Um, we began prospective natural history studies of MPS 1, 2, and 6, and now MPS 4 as well, and adjunctive studies of MPS 3. In 2015, we found in the effort to treat brain disease, the first trial for intrathecal, that is into the spinal cord enzyme replacement, had negative findings for MPS 3. And then in 2015, there was an FDA workshop on assessment of cognitive outcomes. In 2016, the first consensus conference, and then this year, the second consensus conference on cognitive endpoints. So, the current issues in cognitive outcomes is, what are the best methods for treating brain disease in neurodegenerative conditions? Um, there, um, there are many clinical trials, as I said before. Um, there's hematopoietic cell transplantation, which is either bone marrow or umbilical cord blood, which is a standard of care now for MPS 1. It is not effective for MPS 2 and 3, as I said before. Intrathecal ERT, no published results so far, and many trials of gene therapy, protein fusion, and so forth. So these are under clinical trials at this point, except for transplantation, which is an accepted treatment. Why is this so hard? And why is it that we haven't gotten treatments before? It's because you can't cross the blood-brain barrier with large molecules. You need methods such as protein fusion or gene therapy in order to get enzyme into the brain. And the other problem is that we can't find children early enough for effective treatment because the damage is irreversible. Newborn screening is coming online now, but one of the catch-22 situations is you can't get newborn screening until you have a treatment found. You can't find young enough children for effective treatments. So those kinds of things are being dealt with right now in the field. So the current issues in cognitive outcomes in clinical trials are finding sensitive measures of change. Um, we can do this through natural history studies. Um, it's very difficult to do well. It's very expensive. And partnership with industry is critical for obtaining this data because they're the ones with the deep pockets. So they have been very helpful in collecting this data and making sure that uh, you have good training and careful standards of assessment. Um, and one of the problems is that historical comparators may not be acceptable to the regulators. And so one of the things that's happening is that many times there are repetitions of these natural history studies. Um, and the metrics that we use are really insufficiently robust at this point. For example, age equivalent scores, or maybe they're not sensitive in those children who fall below the floor of the test. There are also many design issues, small samples, no possibility of placebo, and there's really a need for international recruitment to get samples that are large enough. And the final thing is that it's a burden on family and examiners um, having to come back on a regular basis. So I want to stop here by having you take a look at this graph. You'll see it again. It really shows you how the neurodegenerative diseases work, where you have a vector of normal development and a disease vector, which has a resultant of disease slowing, plateauing, and decline in those children who have cognitive decline. So I will stop here and I will encourage you to look at the set of references at the end of this slide at your leisure. Thank you.